So welcome back. This is the third and final video of our class on mitochondrial oxidants and antioxidants. And here I want to talk about how to measure mitochondrial oxidant release. And I want to start by talking about some things you have to think about in animal models and in cell models in terms of mitochondrial function and oxidant production that are often overlooked in studies that involve mitochondrial oxidants. The first I mentioned briefly before is that some animal models, and specifically the C57 Black 6J mice from Jackson Laboratories, do not have the mitochondrial transhydrogenase, or the NNT, function. They've actually had a spontaneous mutation with a loss of function of this protein. And this has been perpetuated over decades unknowingly to the people who work with these mice, and we actually found out about this more recently. So these mice don't have the ability to produce NADPH from NADH within their mitochondria. And that, of course, changes mitochondrial redox state, changes mitochondrial antioxidant function, and has a lot of effects on the animal's well-being, actually. And it has been found that these animals are quite prone to metabolic diseases because of this mutation. So in the last few years, the Jackson Laboratories have become aware of this. A number of papers have studied this fact, and they have developed actually mice that don't have the NNT mutation versus mice that do. And you should be aware of if your animal has this mutation or not when you're studying animals. What are the consequences of not having NNT in mitochondria? So these animals have low NADPH and low glutathione in their mitochondria, and that's expected, low reduced glutathione, because they can't reduce NADPH from NADH. They also are more sensitive to additions of exogenous hydroperoxide or organic peroxides, as you would expect, because they have less peroxidase activity. And finally, they can take up less calcium. They're more prone to the mitochondrial permeability transition, again, because mitochondrial permeability transition involves oxidative damage to the inner mitochondrial membrane. So these are animals that are not good models for what a human normally would be in terms of mitochondrial redox state because of this spontaneous mutation. Just as an illustration, this is a measurement of NADPH fluorescence. As we spoke before, NADPH can be measured by fluorescence. And it's measured in animals, the J animals here, in mitochondria from animals that don't have NNT activity, or the U animals here that do have the NNT activity. And they're adding here tertiary hydroperoxide, which is going to oxidize NADPH, as an organic peroxide will do. And what you can see is that animals that have the NNT can recover this reduced state of the NNT very quickly, while animals that don't have the NNT, that rely only on enzymatic production of NADPH in their matrices, take a lot longer to recover these NADPH levels. So there is quite a difference between these animals. Of course, this doesn't mean that you have to negate all the studies you've done in animals that have this deletion. I just really recommend that when you're publishing a paper, you put the exact animal type that you worked with in your methodology. You state if it is an animal that has the deletion of the NNT or not. And also that you take care that when you're comparing animals, when you're comparing knockout animals, for example, that the background of both your control and your knockout is exactly the same, because there are lots of effects of NNT deletion which are really interesting in terms of redox and metabolic effects. And you might not even know if your control has or not the NNT, so check that out. Another point I wanted to talk about relates to cell studies. So there are many characteristics when you study a cell, a cultured cell, that are quite different from a cell in vivo. And one of the important ones is oxygen concentration, and that really changes oxidative phosphorylation and specifically changes mitochondrial oxidant production. So solubility of oxygen in culture media in normal temperatures is around 200 micromolar. However, most of our cells within our tissues, because they're not directly linked to our blood, because there are many barriers between that, and because diffusion of oxygen actually isn't all that good in vivo, most of our cells grow at around 1 to 30 micromolar. So most of the mitochondria within cells, within animals, actually work with much less oxygen than the cells that we grow in our cell cultures. And this is very important because oxidant production in mitochondria is strongly determined by oxygen concentration. And this was very clearly shown by Paul Brooks's lab, who quantified hydrogen peroxide release from mitochondria. And here again, they're using Amplex Red linked to HRP, just like the fluorescence traces I showed you when we talked about reverse electron transport. And what they saw was that with many different substrates under many different conditions, as you increase oxygen concentrations, you have an increase in oxidant production. And very often in these concentrations that you expect a cell to be in real life, you actually have quite significantly less hydrogen peroxide release than you have in the 200 micromolar oxygen that we have in our cell cultures. So whenever you're studying a cell, remember that the cell is being incubated under much more oxygen than a real-life cell probably is, and think if that's important for the question that you're studying. Another point that you have to consider when you're studying cell cultures is that cell lines are grown with very high glucose. And specifically for lines that are growing for a long time, this can select cells that actually use mitochondria less than cells used within an organism. So they're much more dependent on glycolysis because they have this infinite supply of glucose there than dependent on oxidative phosphorylation. There are many cell cultures that actually use less oxidative phosphorylation than the original cell did where it came from. So you can have this decrease of oxygen consumption that's artifactual from the kind of media that we grow our cells in. You can grow cells in non-fermentable substrates, so without glucose, and forcing these cells to use more of their mitochondria. And if that's important for you, that's something that you might want to consider. So how do you measure oxidants from mitochondria? In permeabilized cells or isolated mitochondria, a good solution is to measure release of hydrogen peroxide from these mitochondria using extra mitochondrial probes, such as Amplex Red plus horseradish peroxidase, which I showed you traces of earlier. This is pretty linear, it's quantitative, you can calibrate it, and you can also use pretty good controls, such as adding catalase to remove hydrogen peroxide and seeing that your fluorescence really decreases. There are a few artifacts involved in this that you do have to be careful with. So anything redox potentially could affect your measurements. So you should calibrate your measurements in the presence of all your other reagents and not just mitochondria. Now, measuring oxidants in cells and intact cells is what most people want to do, and that's a lot trickier. So by far the most used measurement of oxidants in cells is probably DCF, and DCF accumulated within the cells. So DCF used as DCF-DA, which is going to be accumulated within the cell. And the reason why DCF is so widely used is that you can measure over time an increase in fluorescence of DCF, and DCF is known to be oxidized and increase in fluorescence by oxidants, so people believe that this increase in fluorescence that they measure is an increase in oxidants. And it's actually very easy to obtain an increase in fluorescence in DCF, which is the reason why I think people like it so much. It's friendly because you can get this increase in fluorescence, which is quite satisfying. The problem with DCF is that it's not specific for any specific oxidant. It could actually be oxidized by many different things, and not only oxidants, as we will see. Another problem with DCF is that as it's oxidized, it can generate oxidants, including superoxide radicals. And that kind of makes it autocatalytic. And if you notice, most traces of DCF fluorescence actually have this ever-increasing rate of oxidation and increase in fluorescence. This does not reflect the production of oxidants in the cell, which probably would be linear over time, but instead reflects the fact that DCF is oxidizing itself from its oxidized form. DCF, in addition to reactive oxygen species, can be oxidized by heme proteins and metals such as iron. So keep that in mind if you're studying something in which you have changes in heme protein levels or you have changes in iron in the cell. And finally, and this is very important, fluorescence with DCF does not correlate with the concentration of the oxidant. So people often show percent increase in DCF fluorescence as percent increase in ROS. And that's wrong because you don't have a one-to-one stoichiometry between the increase in fluorescence and the increase in oxidants. So be very careful with that. If you want to learn more about the use of these fluorescent probes to measure intracellular oxidants and the problems with DCF, there's an excellent review written by many very important authors in the field of redox biology, which I can recommend to you. Specifically for studies that involve mitochondrial changes, DCF has another problem that I really want to stress here because it's metabolically relevant, which is that DCF is very sensitive to changes in pH. So DCF fluorescence, and this is the oxidized form of DCF, and its fluorescence is being measured at different pHs here. There are no oxidants being added. This is completely in vitro. It's just fluorescence versus pH. And what you can see is that within the range that intracellular pH actually can change, so within the range of 6.5 to 7.5 here, you have a very large increase in fluorescence of DCF, irrespective of its oxidation, just because of the change in pH. Now, when you're measuring things that involve changes in mitochondrial bioenergetics, they often involve changes in inner membrane potentials, changes in oxygen consumption, and changes in glycolytic rates and lactate production rates. So changes in pH within the cell are common. And these changes in pH alone can change DCF fluorescence. So if you're going to measure something, proceed with a lot of caution if you're going to use DCF, and always remember that you should never use this probe in isolation as a measure of mitochondrial oxidants or mitochondrially generated oxidants. You need more evidence than just the fluorescence of this probe. Another group of probes that are often used to measure mitochondrial oxidants in intact cells are cationic probes that are accumulated in mitochondria, oxidized, and then fluoresce. And there are a number of these probes: dihydrorhodamine, mitotracker, red ROS, mitosox. All of these are accumulated in mitochondria, and once they're oxidized, they generate fluorescence that can be measured. These probes, you have to remember, will fluoresce dependent on oxidation, but also on their accumulation in mitochondria. And accumulation in mitochondria is going to depend on the inner mitochondrial membrane potential, because these are cationic probes that are going to be accumulated in a manner dependent on the membrane potential. Importantly, their accumulation is not only dependent on the inner mitochondrial membrane potential, but exactly as we saw for membrane potential probes in intact cells before, their accumulation is also dependent on the plasma membrane potential. So you have two membrane potentials that, if changed, will change accumulation of the probe and change oxidation and fluorescence of the probe, possibly independently of changes in mitochondrial oxidant production. So keep that in mind. One thing you can do to sort of control for that problem is use a probe that's not redox sensitive, so it doesn't fluoresce in the presence of oxidants, it fluoresces in general, to control for these different membrane potentials. So you can normalize it to a membrane potential sensitive probe. That solves the problem of accumulation because of membrane potentials. It does not solve the problem of fluorescence because of quenching, which also depends on accumulation. So once again, if you have a lot of probe in the mitochondrial matrix, instead of an increase in fluorescence, sometimes you can have quenching of this probe, and this can decrease the fluorescence. So it's really important when you're using these kind of cationic probes to know if you're working in a range of concentration in which you have quenching, or if you're working within a lower range of concentrations in which you don't have quenching. And in addition to the quenching effect, accumulation in mitochondria also could be associated to damaging properties of these molecules that are in very high concentrations in mitochondria. They may inhibit respiration, for example, or uncouple mitochondria. Finally, as we said with membrane potential measurements also, changes in size and shape can affect the fluorescence of these probes. And to add another insult to injury, another thing is that some of these probes, if mitochondria lose their inner membrane potential, they can actually be released from mitochondria. They don't accumulate stably in mitochondria. And that means that if you have damage to mitochondria and loss of membrane potential, you could have a decrease in mitochondrial fluorescence because these probes were released to the rest of the cell, but that doesn't mean that there wasn't oxidation of these probes. So again, they can be interesting if you use them in addition to other techniques to see what's happening in mitochondria. But these sorts of probes, when used alone, they really cannot be trustable measures of mitochondrial oxidant release. And they're definitely not quantitative measures of mitochondrial oxidant release because you have all these problems related to accumulation and lack of correlation between fluorescence and oxidation. A much more attractive way to measure mitochondrial oxidant production by putting a probe within mitochondria, accumulating a probe within mitochondria, is to use a probe that you can then remove from your biological system and quantify outside of this biological system. Because that eliminates all those problems related to accumulation, related to size and shape, related to aggregation and quenching. And a commercially available system, which I think is quite elegant to do that, is the MitoB, MitoP system, which is a molecule that is oxidized by hydrogen peroxide, but accumulated in mitochondria because it has a cationic characteristic. It produces MitoP, and then you can isolate both MitoP and MitoB from your system and see the relationship between the oxidized molecule and the non-oxidized molecule outside your system, and therefore quantify at the same time how much probe was accumulated and how much probe was oxidized. And I think that's a really much more trustworthy way to measure mitochondrial hydrogen peroxide production in situ. And it's really interesting because you can use it in cell cultures, but you can also use it in animals. So you can use it in Drosophila, for example, to quantify mitochondrial hydrogen peroxide production. A large group of probes for mitochondrial oxidant production are also encoded, genetically encoded probes. There are many different fluorescent probes, green fluorescent probes, yellow fluorescent probes, with disulfides mainly, that were generated to be sensitive to oxidants. And some of them are more selective to one oxidant or another. So they're actually quite interesting in the sense that they could distinguish between different oxidants to some level. They're also interesting because you can target these probes to specific regions. So you can actually have a probe in the mitochondrial matrix versus the intermembrane space versus the cytosol and look at oxidants in these different compartments. The best of these probes are the ratiometric probes because then you have a control for the expression of these probes. If you don't have a ratio measurement, you don't know that in your different experimental conditions you might have different expression of these fluorescent proteins. So that's something important to know. And the best of all of those are those that you can isolate from the cells, these proteins, and then in a Western blot separate the oxidized and the reduced form. And that allows you to quantitate these probes without looking at their fluorescence in situ. So also eliminates all the problems of fluorescence in situ and the interference of all these biological systems in fluorescence. Many of these probes are very interesting, but there are a few problems with them too. Remember that not just because somebody said that they developed a probe to measure oxidant X doesn't mean that really they do measure oxidant X. For example, CPYFP was shown by some authors not to respond to superoxide radicals, while other authors actually believe that they are a superoxide radical probe. Hyper is a probe often used to measure hydrogen peroxide. I think it's a very interesting probe, but it is, as are almost all fluorescent proteins, pH sensitive. And as I said before, many metabolically different conditions involve changes in pH in a cell. So keep in mind that pH might be responsible for changes in fluorescence of these proteins and not just oxidant levels. Finally, remember that any redox-sensitive probe may generate oxidants, especially when it's photostimulated, when you're measuring its fluorescence under a fluorescent microscope, for example. So these probes are interesting, but again, they have some problems, and you always have to keep in mind that when measuring something as reactive oxygen species, you will have more artifacts, and therefore you do have to be more careful. So how can you measure mitochondrial oxidants better? You can join more than one experimental approach in your measurements. And the thing you can add to your measurements is the effect of a mitochondrial antioxidant. So if in addition to measuring some kind of oxidant being produced, you see that your biological effect is inhibited by a mitochondrial antioxidant, or that it happens when you add a mitochondrial oxidant, for example, that's a really interesting way to add a new approach, a new experimental approach to show that your effects are related to oxidants. The most classical mitochondrially targeted antioxidants are ubiquinone targeted to mitochondria in the form of MitoQ, and plastoquinone targeted to mitochondria in the form of the SKQ molecules. These were developed originally by Mike Murphy's and Vladimir Skulachev's groups. These have been used for a number of years. They are very potent molecules in a number of different experimental conditions. They have lots of biological effects, and they're actually being tested in clinical trials for some diseases. So very interesting mitochondrial antioxidants. Now, just using these antioxidants doesn't mean that this process involves mitochondrial oxidants. I would like to stimulate everybody who's using these mitochondrial oxidants to consider also as controls, first using the antioxidant portion of these molecules without the mitochondrial target. And that's a way to see if the targeting to mitochondria is the reason that you're inhibiting your effects, or if it's just the antioxidant itself. And second, and this is really important, to use the mitochondrial target component, that's usually TPP, separately from the antioxidant and see if it alone doesn't inhibit your process. And this is important because accumulation of a cation can lead to mitochondrial uncoupling, and mitochondrial uncoupling has a whole bunch of metabolic effects, as we saw before, including decreasing mitochondrial oxidant production. So you want to make sure that the effect is because of the antioxidant and not because of the mitochondrial target itself. But having said that, doing those controls, use of mitochondrially targeted antioxidants is a really interesting thing to do. So we saw that we have a lot of problems when we're going to measure mitochondrial oxidants, and that's not something I would expect because oxidants and free radicals in general are very reactive. So it's going to be difficult to measure them very precisely within a complex biological system. So I want to give you a few tips on how to measure mitochondrial oxidants without too many artifacts. I'm going to call them foolproof, but I don't think there's anything foolproof in anything in science. This is just prevention of most of the problems that you can have. Because of course you do want to measure mitochondrial oxidants. You don't want to give up on doing it just because it's not a simple thing to do. So my recommendations in terms of studying mitochondrial oxidants are, first of all and foremost, please do not use a fluorescent probe in isolation and say that an increase in X% of fluorescence means an increase in X% in concentration of whatever oxidant you think you're measuring. We saw that these fluorescent probes typically are not specific for one oxidant, and we saw that these fluorescent probes do not have a one-to-one increase in fluorescence versus the concentration of these oxidants. So don't normalize and say percent increase in ROS with any one of these probes. Be honest. Say percent increase in fluorescence of X probe. That's what you measured, so that's what you can actually say that happened. Remember that all these probes and all the fluorescent proteins also tend to be non-species specific. So even though sometimes they're sold as being specific for superoxide or hydrogen peroxide or peroxynitride or whatever, usually they're oxidized by more than one species. It's just a question of affinity. So don't really rely just on probes to know what the specific chemical species that you have. Always think, if you have changes in pH, if you have changes in membrane potential, if you have changes in expression of encoded proteins, or if you have changes of size and shape in mitochondria, that may be a reason for your changes in readout. So always consider that as a possible confounding factor in your measurements. And never use a single technique. So the thing about redox studies, because there's so many artifacts, is you have to use more than one technique, and preferably more than one experimental approach. So what I really recommend is a combination of redox strategies, using at least 3 different approaches to solve your redox problem. One approach is to try to measure your oxidant, either with a real-time measurement, encoded probes, fluorescent probes, or with a time point measurement such as the Mito-P, Mito-B system that I showed you. Most people like these measurements over time. There really is no reason to do time scans in most studies. It can be a specific time point measurement. I think it's just a psychological thing that most of us like to see this increase over time. It's not necessary to do in most experimental situations. The second combination that I really recommend is to measure some kind of oxidation product in your sample. So to measure oxidation of proteins, of lipids, of DNA, a modification of a protein that you expect to see within your sample with the production of the oxidant that you expect your sample to be producing. This is a second measurement of the presence of this oxidant and really is going to make the reality of this oxidant there more trustworthy. And finally, try to see if your biological effect that you're relating to these oxidants can be modified either by antioxidants, removing these oxidants, or by adding in oxidants. So if you can have your biological effect by adding in externally the same oxidant. And these are really 3 different strategies to look at the effects of oxidants in your system. And if you have all 3 of them together, I think you'll have a really convincing story about how oxidants work within your biological system. So that's the last class of our mitochondrial bioenergetics course, and the last theoretical class, actually. I'll see you next week in our best class of all, which is going to be the class in which we're going to talk about your bioenergetic problems. You're going to bring your studies or your ideas for projects within mitochondrial bioenergetics, and we're going to talk about how we would approach these or how we would tweak the experiments that you plan to do to have even better results. So I'll see you then.